Unexplained bee deaths in the spring of 2014 all but wiped out thousands of hives in the Coromandel, Raglan and Wairarapa areas. Dr Mark Goodwin from the Honeybee and Pollination Research Team at Plant and Food Research is trying to determine the role of 3 gut-dwelling parasites in the bee deaths.
We got a phone call last spring from a beekeeper in the Coromandel Peninsula who reported that a large number of their colonies were dying. And the symptoms they described to us were symptoms we hadn't heard of. So we went up and had a look at them, and they were certainly new to us as well. What started with colonies in the spring with about 15 or 20,000 bees, which would normally grow up to be really big colonies and produce honey, when the beekeeper came back about a month later, all there was a queen and about 200 bees. And it's not just one colony, we're talking hundreds and possibly thousands of colonies. We organised a big beekeeper meeting in the Coromandel to find out how many people were affected. And when they turned up, there was a whole lot more who had losses who we didn't know about because they assumed it was something they'd done rather than something more generic. The symptoms look like what we see overseas that are loosely described as colony collapse disorder, which is a problem in Europe and through the US. Just because the symptoms look like it doesn't mean that's what the cause is though. When we tested the bees, they had very high levels of a couple of gut parasites. Nosema apis, which is a microsporidium parasite, which has been in New Zealand forever, and it's very hard to associate it with these symptoms. But we do have a new one that was first found in New Zealand in about 2006. It's Nosema serrana, and it's an organism that a jumped species from an Asian species of honeybee, the same place Varroa came from. They also have Lotmeria, which is another well, we don't even know if it's a pathogen. It's a new record for New Zealand being present here, but it may be just that we've never looked before, and it may have been forever as well. And we know that these things are in very high levels, and we know for Nosema apis and Nosema serrana, at least high levels usually mean bad things for colonies. Whether it's the cause of everything we're seeing here, we just don't know yet. Lots of things have been suggested to be the cause, ranging from cell phones, through to neonicotinoids, through to pollen shortage, through to pathogens. In fact, everything you can imagine has been suggested as a cause for it. In reality, we don't know. When we started the work, there was a few things we could exclude straight away. A lot of the world has pointed towards a class of chemical neonicotinoids as a potential cause for colony collapse disorder. But we're working in the Coromandel, which is mainly regenerating forest. There's not a lot of chemical use at all. So it was very unlikely that that was the cause. When we're looking for hypotheses to explain why these colonies have all these gut parasites suddenly, there is something else, of course, that's happening out there with bees in New Zealand, and that since 2006, we've doubled the hive numbers in the country. We've gone from 260,000 colonies to 500,000 colonies. And they've been put mostly into very high stocking rates on Manuka. So we've got densities of bees that we've just never seen before. Whether this has some relationship with what we're starting to see at the moment, we don't know. The symptoms are in places where we do have high densities. We can't conclude from that that's the cause, but it does point us in a direction that we certainly going to look at. And particularly what happened last autumn about food availability, whether they all had enough pollen going into the winter because of this competition, is a question that is sitting out there to be answered. We've just started with these samples of bees that we know have got high levels of disease. We know nothing else about those bees other than what hives they came from. And the bees that left the hive, we can't even sample. So what we need is colonies with bees that we can track, that we know when they were born, we know how long they've been in the hive, and we can sample at different times to work out their disease loadings, whether they leave or not, and whether it's affected their lifespan. And that's what the marking of the bees are. So we can have a lot of colonies with bees that we can actually recognise. We take frames out of a colony, shake all the bees off and put it into an incubator. And then every day new bees will emerge from the colony. We can pick those off and we can mark them. Really great thing about using cold-blooded animals like bees is if you cool them, they stop moving. So we put them in a fridge for a few minutes, they stop moving, we can paint them and warm them up again, and they just carry on as normal. They're great to work with for two reasons. One is they don't sting you, But a second one is we know they don't have any other diseases. They emerge almost as a clean slate. So any diseases they get now, they'll get from the colonies we put them in. So when we come back and sample them, we can know a lot about the colony we've put them in and their history in there to do the testing. Worldwide, there is one treatment available for Nosema, and it's fumagillin. However, it's banned in a lot of countries because of the residues and the product that it is. It's not allowed in New Zealand, and it's not allowed in Australia. And so we actually don't have anything present at this point in time that we know will definitely work against Nosema. There are some organic products that have some claims that way, but it's really only guessed to see whether it's an effect. So that's one question we're having to go through now, is the permission to, one, get to use it for experimental purpose, but then the industry is going to have to answer the question, assuming Nosema cerana is the issue here, do they want to have a chemical treatment that they can use? Planted Food has put some of their core funding into this problem, and we're just talking to all the people in New Zealand who actually use bees, either to produce honey or to do pollination, to come in for co-funding to help fund the project. Regardless of all of that, we've started investigating it, and we'll just assume that the rest of the funding we need will turn up to actually finish the project. It will be really interesting for us as researchers and for the regulators who are keeping an eye on things that are happening if beekeepers do report where they have losses. And there are a number of ways of doing that. The Environmental Protection Agency has a website that you can report directly onto. Secondly, beekeepers can report through the National Society. And also the National Society can arrange from a commercial lab in the Hawke's Bay to get samples tested, which the results come back to us as well, which is very handy. So again, part of the research question is looking at hives individually, but also get a feel overall what's happening. And that sort of reporting is actually very useful.
This program was made with funding from New Zealand On Air.